Previously, we talked about using a N protector on an amino acid because if we want to make a protein, then we need to have some control over how these amino acids begin to hook up. So I take an amino acid, I put a N protector on it, and this covers up the nitrogen of the molecule. Now I want to kind of make the C terminus of that amino acid more attractive. I want to make sure that the molecule will hook up in the order that I want it to hook up in. So what I do is that I take a look at the nitrogen and I protect it, and then I take a look at the carbon and I activate it. So again, the template for an amino acid is NH3 carbon with a hydrogen and a COO minus, right? A C double bond OO. So what I want to accomplish here is protecting this group, which is the head. That's the N-terminus. So that N-terminus, I can go through and say, I don't want you to touch it. I'm going to cover it up, and you're going to pretend like it's not even there. And then I'm going to go through and I will look at the C-terminus end and I will activate it. So that means I want to make it more attractive. This is the order that I want it to hook up in. We talked about the reagent that was needed in the previous video of how to protect the NH3 group. So what reagent is needed to make this COO more attractive? Well, again, we call this the C-terminus activator. And what we use here is another long drawn out term. So it's dicyclohexyl carbo-diamide. And we do give this one an abbreviation. We call this DCC, so dicyclohexyl carbo-diamide. Now, what would this structure look like? Well, that name is going to tell you what that structure looks like. We have a diamide. So an imide, if you remember, is a carbon that's double bonded to a nitrogen. Well, I have two of those. So what this structure looks like, well, it starts off, it looks something like this. And then I've got dicyclohexyl. So that means off of this nitrogen, I have a six-membered ring, and off of this nitrogen, I have a six-membered ring. So that's the structure for dicyclohexyl carbo-diamide, otherwise known as DCC. And this molecule will go in and activate the carbon. It makes a very good leaving group in the mechanism. So when this begins to form and when this begins to go on to the C-terminus, you end up with a very good leaving group in the very end, bottom line, that can easily be replaced and activated, and something else can come on into its position. So that is the C-terminus activator. We call it DCC. So what would happen here? Let's say that I had two amino acids. Glycine is one of them and alanine is the other. And let's say that I want to go in and I want to make sure that this hooks up glycine first, alanine second. So what would happen is that I would take a solution of glycine and I would add my N protecting group on it. So what I'm going to do here is I'm going to put N star. So N protector group is going to be added on to glycine. So this was using our previous reagent of ditert butyl dicarbonate. Then on this glycine, I would also add DCC. So DCC would also be added here. So I've taken this glycine and I've now said, okay, I want glycine first, so I'm going to protect the N group to make sure that's N-terminus. And then I want to make sure that the glycine will come and actually hook up with a peptide, and I'm going to activate that C-terminus to make it more attractive. So what happens at this point is that alanine, that hasn't been touched at all, will come into the mix, and it will say, oh, there's DCC there. That's very attractive. I think I'll swoop in for the kill. And we get glycine now that still still has this N-terminus protector group on it, but alanine has now joined up with glycine. It's kind of came in, taken a look at DCC, kicked it off, and then alanine has taken the DCC place. Almost kind of like a substitution reaction. So here we get glycine that comes first, and then alanine that has come in second. Now, typically what I would do if I wanted to continue on with maybe a third amino acid or a fourth amino acid is the next step. So the first step here, you add your N-terminus protector on. In the second step, you will add your C activator on. In the third step, you will then add your amino acid that you want to come in second. In the fourth step, you would do a cleanup process here. So you would take this dipeptide, you would extract it out of solution, and you would have this dipeptide of glycine and alanine. Then you will turn around and you will add another C activator onto the molecule. So that means that at this point I could then add DCC again, and this time it would be alanine that would be activated because it's the last one in line. It's the one that has the free carbonyl now. So the DCC would go on to the second amino acid. I would then throw in a third amino acid. So let's say histidine. So histidine will come in, kick DCC off, and now I've got a tripeptide in the order that I wanted it. So then I would have to do the cleanup process again, and then I would C activate it again. So these two steps, you wash and repeat, just like the shampoo in your hair. So wash and repeat, follow the directions. It's for that reason that this process becomes very unreliable the bigger the protein gets. So if you're working with small proteins, then this technique is okay to use. But if you're working with proteins that have a number of different amino acids, you're actually going to see that this is going to get harder and harder and harder to accomplish. Actually, I think the statistics show that if you bring in 9 amino acids, 9 of them, well, your percent recovery here is going to be about a 15%. So 15% of all the hard work that you've put in to this protein assembly, you'll actually get back as the protein that you want. And it's due to all of the cleanup, it's due to all of the washes, it's due to all of these complications that can happen in the synthesis portion itself. So that's the issue here. When you get finished, we do have a very last step. And here I'm just going to put a star because it really depends on how many steps of cleanup and C activators that you have. So the very last step, we need to remove this nitrogen protector. And we do that by using another reagent. Now the nitrogen protector does go by a name, and this is called TBOC. It stands for tert butyl oxycarbonyl. So TBOC, so when the desired number of amino acids has been assembled, let's say that you want 15 amino acids, and you've assembled these 15 amino acids in this way. You protected, you activate it, you brought in the second. You activate it, you brought in the third. You activate it, you brought in the fourth, on and on and on and on. When you are finished with the assembly, well, you've got to get rid of the protecting group. And again, the protecting group is called TBOC. So the way that you do this is that you use trifluoroacetic acid. And the dry fluoroacetic acid, and it needs some solvent. Typically the solvent's DCM, dichloromethane or methylene chloride. It won't break any bond except for the N protecting bond, and we get some byproducts that come from it. If you want to know what this reaction looks like, it's in the text. You can kind of read it. It will show you the mechanism that's there. Again, that's not something that we're going to be focused on, but you do have to remove that N-protecting group. That way you can have a true N-terminus on your protein at that point. So once you get this assembly, you will then have to go in, and let's say that we've ended up now with an N-protecting group on glycine, and then alanine, and then histidine, and that's kind of where we want to stop. Well, I would use trifluoroacetic acid and DCM, and what goes on is that this will get removed, and now we got a true protein of glycine, alanine, and histidine. Now notice I deleted the nitrogen. The nitrogen stays. I only put the N there to let you know that this was the beginning of the protein and that was protected. However, in this case, the nitrogen is still involved in the amino acid. It's still now really an NH3+. It just doesn't have a protecting group on it. So trifluoroacetic acid and DCM will go in, remove that protecting group, and convert it back to that N-terminus of the actual protein chain itself. So that is kind of how we have some kind of control over protein formation and making protein in the lab. It's a very complicated procedure. There is an automated process. It's called automated solid phase peptide synthesis that we can actually do in the lab. It's a little bit better, but still the results of it are as low sometimes as the hand result. The only thing that it saves us is the trouble and the time for doing it by hand, right? It's like a bread maker. Who wants to make bread by hand? Why can't I just buy a bread maker and let it make it for me? Well, that's the purpose of the instrumentation. So there is automated procedures out there. You can find it in research and development. You can probably see it used. Would you ever come across it? Probably not. So that's why I'm just not going to spend a lot of time on it. If you're working with some biologicals, if you're working with some biochemistry labs, however, you might see them have one of these in their lab. But for the most part, many of you will not go that route, and you just simply won't see it at all. Okay, so with that said, in the next video we'll continue on with this idea of what we call primary sequence. That's what this stands for. So primary sequence is basically the order of the amino acids in the actual protein itself. So glycine comes first, alanine came second, histidine came third in this example. So that is the primary sequence of the protein. We'll continue on with this idea of primary sequence in the next 10-minute clip, and then we'll just continue to work from there. We're almost finished with amino acids. There's a couple of more maybe complicated examples that we have to work through, but as far as the theory, we're almost there.